This video covers Advanced Higher Biology, Unit 2, Organisms and Evolution, specifically Key Area 1, Field Techniques for Biologists. We're going to look at monitoring populations, which is subsection D, describe the use of indicator species to monitor the environmental conditions in an ecosystem, describe the process of mark and recapture, and talk about how it can be carried out in an ethical manner. The first section of this video acts as a reminder of the content covered at National 5. We as biologists often monitor populations in order to understand which conditions are occurring in the ecosystem. Presence, absence, or abundance of the indicator species can give information of environmental quantities such as the presence of pollutants. For example, biodiversity of lichen species in an area can indicate the levels of sulfur dioxide in the air. Likewise, freshwater invertebrates are frequently used to indicate sewage pollution. The absence or reduction of populations indicates that a species is susceptible to the environmental factor, whereas if we find they're quite abundant or if there's an increase in the population with the change in the environment, that indicates they favour those conditions. Therefore, we can use susceptible and favoured species to monitor an ecosystem. To effectively use the abundance of these indicator species, we need to first know what their population sizes are. A sample population is sampled and marked and then released. So on the first day, we captured and marked 2, 4, 5, 10. So this is our M value, so for the initial marking. After an interval of time, so that's decided by the researcher, probably in a pilot study, we look at a reasonable amount of time. A second sample is captured and recounted. This gives us our C value. The individuals within that second sample, which have been recaptured, so still have the red dot that the original capture group had, gives us our R value. So we then do the number in the population as our estimate is marked initially multiplied by captured, the second lot, divided by the number that were recaptured. Whenever we make an estimate, we must also make assumptions. In this case, we assume that all individuals have an equal chance of capture, so it might be that very fast individuals just are unlikely to be captured, that there's no immigration or emigration, so if you imagine some of these red turtles left the area and were not available to be recaptured, that would alter our R value. And we've got to assume that those which are marked are able to mix fully and randomly within the total population. So if after we capture an animal, it's then traumatized and doesn't move at all until we then do the recapturing stage, you're going to definitely catch it in your second group. So we have to make sure that they're able to mix fully back into the population after our initial capture. There's multiple different methods we can use to mark individuals. We can use banding, tagging, surgical implantation, painting, or hair clipping. The method of marking and subsequent observation must minimize the impact on the study species. If through tracking we cause the animal to have a reduced fitness, this is deemed to be unethical. This could be in terms of natural selection, where the impact on hunting in a predator or evasion in a prey species is decreased, or in terms of sexual selection, if the tracking caused an increase or decrease in sexual function or attractiveness. This would be deemed to be unethical. So, presence or absence of an indicator species can give information of environmental qualities, such as the presence of a pollutant. The absence or reduction in the population indicates that that species is susceptible to the factor that's being measured. Abundance or an increase in the population indicates that it is favoured by the conditions. A sample population is sampled and marked and then released. This gives the M value. After an interval of time, a second sample is captured and counted. This is C value. If some of the individuals in the second sample are recaptured, these again are counted as the R value. The total population is estimated using the equation N equals MC over R. This method assumes that all individuals have an equal chance of capture, that there's no immigration or immigration, and that individuals are, once marked, able to mix freely and randomly within the population. In marking, we can use banding, tagging, surgical implantation, painting, or hair clipping. The method of marking and subsequent observation must minimize the impact on the study species.